the fat in blood. Does anybody know what fat in blood is? It's called triglycerides. So you're eating a whole bunch of fat, but you're not eating carbohydrates. What happens to triglycerides, the fat in blood? It goes down or goes up? Goes down, okay? Because the body now is forced to burn fat, so it doesn't leave fat energy hanging in the bloodstream, it clears it. Now, what happens to the good cholesterol? You know, when you're eating a large amount of animal food, so you're burning fat, what happens to your good cholesterol level? Does it go up or down? It goes up. What happens to your blood sugar levels? They go down. What happens to your insulin levels? Do they go up or down? They go down. What happens to your inflammation markers? The markers that cause, they go down. So you are getting a lot of benefits. Blood pressure is down, weight is down. But what is the one single factor that happens that worries everyone? What happens to the so-called bad cholesterol, the LDL cholesterol? It goes up. So the question that I want to pose today in front of you is that the LDL cholesterol going up, is that necessarily bad? No. Okay. So there are a lot of converts out here who say it's not bad. And today I want to kind of present a lawyerly case, a lawyerly defense of that LDL molecule, saying that that high LDL may not be as bad as you think. In the setting of a low carb diet, cholesterol in our body performs a lot of different functions. So some of the things that physicians forget and people forget is that cholesterol is highly important for our body. Every cell has cholesterol. So if you take a cell, we are composed of billions of cells. The cell membrane, which is the outer covering of the cell, what gives it the structural integrity? What gives the integrity to that cell membrane? What is the molecule that we need to give it integrity? Many things, but one of the principal factors is cholesterol. Cholesterol provides the membrane with the right degree of flexibility, with the right degree of prevention of penetration of bad chemicals into the cells. So the way the shape of your cell, every cell is highly dependent on cholesterol. So we established that there can be no life without cholesterol because the cell membranes would refuse to function. Is that the only function of cholesterol? No. We think, we have cognition. Is cholesterol important for that? Yes, it is. So the membrane of the brain, the way it folds, depends on cholesterol molecule. So cholesterol creates these rafts, which is a structural part of the cell membrane of our brain. And these rafts are important because that's where the neurotransmitters come in and sit at. So neurotransmitters are brain signaling molecules. So these signaling molecules will not work if our brain did not have enough cholesterol. And in fact, cholesterol is so important for the brain that the brain does not delegate the responsibility for making cholesterol to anybody else other than the fact that the brain makes it on its own. About 25% of our body's cholesterol resides in the brain. So we talked two functions so far. We make steroid hormones called cortisol. A presentation in front of a large audience creates stress. Will I remember everything? Will I remember the sequence of slides? Did I do enough homework to come and present in front of you? These are all stressors. How do I counteract with that stress? How does my body deal with that stress? Have you ever thought about that in an emotional situation? That's the adrenal gland that makes it. What is the basic backbone for making of that cortisol molecule? It's cholesterol. Our virility, our muscle mass, our feminine characteristics, our masculine characteristics depend on sex hormones. Sex hormones, what is the backbone for the sex hormones? Where does it come from? It comes from cholesterol. Okay? Specifically, for whatever reason, the ovaries which make estrogens and the testes, which make testosterone, cannot make their own cholesterol. Surprising. And they depend on the LDL, which is the bad cholesterol, to supply it with cholesterol to make the sex hormones. How does the body use cholesterol other than what I told you about? How does it get rid of it? Have you ever thought about that? The way the body gets rid of cholesterol, amazingly enough, our gallbladder makes bile acids. What is their backbone that gives you bile acids? It is made of cholesterol. The liver is making about 5 grams of bile acid, and there is about a 10 to 20% loss of this. And can the liver modulate the loss? Can our body modulate the loss? Like, let's say I go on a fasting program, and I'll show it to you. In my fasting, my LDL cholesterol or bad cholesterol is going to go up. So if your body is making a large amount of cholesterol, the body will eliminate more cholesterol because it doesn't need it. This is an important factor. Vitamin D is deficient in 70% of Americans. Where does vitamin D come from? The cholesterol in the skin gets hit by the sun rays and it gets converted to vitamin D. So vitamin D is a cholesterol byproduct. What I didn't tell you is that is our muscle function dependent on cholesterol? The reason is that from cholesterol, from the same pathway that is making cholesterol, comes CoQ10. And CoQ10 is the spark plug for the muscles. It's erroneous because it's very simplistic when you think about cholesterol, because when you measure the cholesterol in the bloodstream, you're really measuring what is called lipoprotein. And the reason I say that is that cholesterol is like fat. So if you have a beaker of water and you put oil in it, the oil remains separated, doesn't dissolve. And the reason I put that up is that triglycerides and cholesterol are both fatty substances. They don't dissolve in our blood. Our blood is watery. So since they don't dissolve in the blood, our body creates an ingenious system to carry cholesterol in the bloodstream. So this is what is called a lipoprotein. It happens to be one of the specific lipoproteins called VLDL. But basically it has an outer layer which is composed of phospholipid, which just means a kind of fat. It has got some specific protein markers that identify the kind of cholesterol particle, and the cargo of that is both fat and cholesterol. When you say your LDL cholesterol is high, what people don't understand is that the cholesterol in LDL is the same kind of cholesterol that is present in HDL. It's just a different carrier molecule. Okay, so this is a very important slide, and never get tired of showing this. So here is a group of 10 people, and they are all healthy, and I say for 7 days you go on a fast. You don't eat anything, because our evolutionary heritage is that of fasting. We had to find our food, so we went through days and days without eating. So fasting is inherent in our biology, and if we don't fast, we don't do good. That's why we promote fasting. So these guys went through a 7-day fast, water and salt only, no calories. What do you think happened to the bad cholesterol, the LDL cholesterol? It went up. So as which shows up here, it was 112 when they started the fast, it went to 190. There was a 70% increase. Now out here is called IGF. IGF, higher the levels of IGF, the greater the cancer risk. What happened to IGF levels? They went way down. The total cholesterol levels went up because when you have an increase in LDL, your cholesterol also goes up. And why is that happening? So it's an important question that this person is fasting, he's eating nothing. Why is the cholesterol going up? And it would shock you to know that up until December of last year, I didn't understand it. And it's only after looking at lots of research papers that we came to this conclusion, and now I can say pretty confidently that this is the mechanism. A person who is fasting, so only thing that they are eating is the fat they have stored. Why do I say that? Somebody may say, no. Somebody can store carbohydrate, you can burn the sugar that you have stored, right? How much sugar do you think our bodies can store? 6 hours, 8 hours, several days. So really the realistic answer is limited to about 6 to 8 hours. Beyond that, there's no more sugar. So when you are burning fat and creating energy, this is a complicated biochemical pathway. And the only reason I put it up is that you're taking fat, you're converting it into a small molecule that comes from fat that's being used in the mitochondria to convert to energy. So you make energy and carbon dioxide. But a part of that is being used to make ketones. Why do we need to make ketones? So you're fasting or you're eating a low carb diet. Why do you need to make ketones? The reason you need to make ketones is because the sugar production has gone way down. The brain depends on ketones now for 2/3 of its fuel. So the brain can change from burning sugar to burning 2/3 of its energy from ketones. So the liver has to make ketones. So ketone production goes way up, and ketones come from chemical synthesis of acetyl-CoA through a lot of different pathways. This is HMG-CoA, and then it forms the beta-hydroxybutyrate, which is the principal ketone in our bloodstream. So a person who is fasting, a person who is not using carbs, by design is making a lot of ketones. And this is the part that most people didn't understand, because this diagram that I have put out to you, if you go and search the Medline, does anybody know what Medline is? So Medline is the entire literature, medical literature in the world. And if you search the Medline, and if you are looking for this diagram, you got to go back into the 1940s, because people have forgotten this diagram. And what it is that the enzymes that are involved in making ketones are exactly the same enzymes that make cholesterol. So when you drive up ketone production, you're going to drive up cholesterol production. Do we know this for sure? Yes, we do know it for sure, because higher the ketones, and you plot in those people, higher will be their cholesterol. Does our body need to absorb cholesterol at that point? What happens to cholesterol excretion in that situation? Increases. Somebody said increases. Yes. So there are studies from the 1940s to 2010 that show that in ketosis your cholesterol excretion increases. So the bile you're making, you absorb less of it. The cholesterol that is in your bloodstream gets eliminated through the gut into the feces. Now, the liver has something called an LDL receptor. Do you know what an LDL receptor is? So on the surface of the liver are a bunch of these molecules. These are receptors that soak up LDL cholesterol from the bloodstream. So think about it for a minute. So the LDL cholesterol is removing cholesterol from the bloodstream, but the liver already is making a lot of cholesterol. So does it need to absorb that LDL cholesterol? It doesn't. So what happens to the LDL receptor? Does it go down or go up? It goes down. And there are biochemical studies that show that. So in a sense, when you're on a low carb diet, what is happening to you is that your LDL cholesterol is going up, because that's what biology intended for it to do. It's a natural consequence of going on a low carb diet and of fasting. So inside the mitochondria is a machinery that is converting the fuel that you use in the presence of CoQ10, and the end product of that is ATP. ATP is energy. So you will see that there are ATP molecules that are being formed, because CoQ10 is a part of the mitochondria that's helping it burn fuel to convert to energy. Okay, so here is a rat experiment. They took rats and they divided them into 4 groups. This is a control rat. This is being CoQ10. This is given CoQ10 plus a cholesterol medicine, and this rat is just being given a cholesterol medicine. And what you do is that you weigh the rat to see their muscle mass. Higher the weight, that means they're forming muscle. You put them on a rat exerciser, it's called rotarod, to see how long they can exercise. And you do certain blood tests to see if there is chemical damage to the muscles from the use of cholesterol reducing medicines. Okay, so this is muscle mass. So in other words, if the muscle mass is going up, the rat is increasing in weight, and this is the control group and the CoQ10 group, they increase their muscle mass. But when you give them cholesterol medicines, at 21 days, their muscle mass is dropping dramatically. Okay. Is there evidence of chemical damage from the muscles? If the muscles get damaged, they release certain enzymes in the bloodstream. One of them is called CK, creatinine kinase. This is the group that is given the statin, atorvastatin, and that's the highest amount of muscle damage at both 14 days and 21 days. This is another muscle enzyme which is called myoglobin, which is again released into the bloodstream. The control group and the group that was treated with statin, a 5-fold increase in evidence of muscle damage. Now what about exercise performance? Did it affect the rat exercise performance? So at 14 days, the only group that had reduced exercise performance was the group that was treated with cholesterol medicine. Then the group, and at 21 days, the highest reduction in exercise tolerance was in the statin group in these rats. What about humans? Is there any evidence in humans? So these are not people with heart disease, these are normal people, 420 of them. They've been given a cholesterol medicine for 6 months, and these researchers who are doing this are paid by the cholesterol industry, so really they should find no evidence of muscle damage. Yeah. But they can't ignore the findings. This is assessing muscle damage in terms of activity. They were given an accelerometer, and the more the counts you had, the more active you were. The less the counts you had, the less active you were. And most of us out here are over 50 years of age. So if you were 55 years or greater, the reduction in your activity was dramatic when you were on the cholesterol reducing medicines compared to the placebo group. So there is evidence from research studies in normal people in a 6-month time frame that there is significant reduction in activity when you take a cholesterol medicine. How long do you think people usually take a cholesterol medicine? 1 year, 40 years? Long time. These are normal people. Was there evidence of liver damage and muscle damage? So this is just a 6-month follow-up. This is not 40 years of follow-up. This is the placebo group. This is the statin group. There was significant rise in the risk of muscle damage and liver damage on the cholesterol group. So you can see that there was evidence of biochemical muscle and biochemical liver damage. Now this is an amazing study. I told you that the brain contains cholesterol, that these are the receptors through which our neurotransmitters work, and out here these yellow things are the cholesterol rafts that stabilize these receptors. So if you don't have cholesterol, brain neurotransmission doesn't function well. So is there clinical evidence that this happens? So there's a town in Edinburgh called Lothian, and this is the Lothian birth cohort in which they had about 1,000 kids that were born in 1936, and they were followed all the way into the 20th century. And what they wanted to do was to see what is their brain function. So you're seeing how good your brain function is in relationship to the amount of cholesterol you have and whether you're using statins or no. So the groups were divided into low cholesterol, which is less than 200, middle cholesterol, which was 200 to 240, and high cholesterol, which was greater than 241. So this is who these people were. Women are singularly lucky. If I take an older person, and if it's a female, and they are healthy, if I take an older person, and if they are female, and if they are healthy, they are very likely to have high cholesterol. Okay? Let me say that again. If you are a female and if you're older and you're healthy, you're very likely to have high cholesterol. So the highest cholesterol was not found in men but in women. Only 30% were men. The highest cholesterol was found in professionals, not the lowest cholesterol, the number of professionals were less. Manual labor was high with low cholesterol group. The higher the cholesterol was there, the lower the chances of you doing manual labor in that 1,000 kids that were followed. What about high blood pressure? People with low cholesterol had high blood pressure. The highest cholesterol people had the lowest blood pressure. People with strokes, the highest incidence of strokes was in the lowest cholesterol group, not in the highest cholesterol group. The highest risks of having heart disease was in the lowest cholesterol group. When I show this slide about stroke and cholesterol, people get so confused. And I don't know why. Because population-based studies clearly show that as cholesterol increases, the incidence of stroke goes down. World War II Japan, at the end of 1940s, had cholesterol levels that were roughly 2/3 that of the United States. The risk of stroke in Japan at that time compared to the US was roughly 5 to 10-fold higher. Not 5% or 10%, but 5 to 10-fold higher. As Japan became rich and affluent, their animal food intake went up. Their cholesterol levels now reached that of the United States. What happened to the stroke incidence in Japan? Did it go up or go down? Down. It fell off the cliff. Their cholesterol levels went down like crazy. So we said we're going to evaluate cholesterol levels and cognitive ability. So general ability is how well they function. The best functioning was there in the people with the highest cholesterol group compared to the lowest cholesterol group. How fast you processed information, highest cholesterol group had better processing speed. They had better memory skills. They had better verbal ability. The higher your cholesterol, the more your verbal ability. You had better ways of formulating sentences and thoughts. IQ at age 70 was highest in the middle and the high cholesterol group, lowest in the low cholesterol group. What about statins? Of the 1,000 people, 300 of them were on statins, 700 were not on statins. Did statins make an impact on cognitive function? Yes, they did. So the statin users are in blue, the non-statin users are in orange. And you can see that in terms of general ability, processing speed, and memory, the people who were not on statins did better than the people on statins in this Lothian birth cohort. Same thing with verbal ability and IQ. Statin users had lower IQ and lower verbal ability. At least with the Lothian birth cohort, I'm giving you a demographic study that shows that high cholesterol, maybe not causal, but is associated with better memory, better cognitive skills, and better overall functioning. But I also told you that cholesterol is involved in infection. The LDL cholesterol, which we malign so much, helps fight infections. So here are a group of bacteria, and when they infect an animal, and here they're trying to infect a rat lung, they release a protein. And this protein is called quorum sensing. So what it's doing, so it releases this protein into the system, and this protein comes back and gives it signal saying that, hey, this is a beautiful host. We can take over and multiply. So it's sending out a signal saying that, hey, I'm going to establish an infection in this area. And then when that happens, the bacterial virulence takes hold, and a lot of bacteria start multiplying. So this is called quorum established. Bacterial multiplication is happening. What is the molecule that helps squash? What does it soak up this protein that is giving quorum sensing to the bacteria to multiply? It is nothing but the LDL molecule. It's sucking up the accessory gene regulator, that protein, and when it does that, it neutralizes the bacteria, And in the process, it ends up killing the bacteria. The bacteria also release what is called inflammation, inflammatory mediators. When I fall off the bike, my hand became red, this was red and swollen. Similarly, the bacteria release LPS and LTA, and these create inflammation, and this is to depict inflammation, and inflammation leads to cell death. What is it that neutralizes these two things? It is the same LDL molecule. The LDL comes in, soaks up the LPS and LTA, reduces the chances of the bacteria getting foothold, and it ultimately neutralizes the bacteria. How many of you guys have heard about a condition called familial hypercholesterolemia? So it's an inherited disorder in which cholesterol levels are about 3 to 5-fold higher. Right? So what happens in our liver is that our liver has these LDL receptors. This is the LDL. The LDL receptor soaks up the LDL and internalizes it and removes the LDL from circulation. So the LDL receptors are taken up by the liver, and the cholesterol and the protein and fat are recycled. Now, familial hypercholesterolemia is a condition in which either the LDL receptor is not there, or if it is there, it doesn't function well, or it's internalized so that it doesn't take the LDL away from circulation. So the amount of LDL, that is the bad cholesterol that we call it, is about 5 to 7-fold higher. So we take mice that have the LDL receptor deficiency. So these mice have high amounts of cholesterol. So their LDL cholesterol is about 10 millimoles compared to 2.5 millimoles in the normal mice. And you give them the bacterial toxin, or you inject them with bacteria itself. And we see which animal dies first. The animal with high LDL cholesterol or the animal with normal LDL cholesterol. And what you see is that the normal LDL cholesterol die very quickly when you inject the mice with the toxin, whereas the mice that have the high LDL cholesterol require a lot more toxin before they die. In the process of neutralizing that toxin or neutralizing the bacteria, you see that the LDL is being consumed. This is what it started out with. After you injected the toxin, the LDL levels fell, and after you injected the bacteria, the LDL levels similarly fall, but there is not as much room in the normal mice. So what this slide is supposed to show is that if you had normal levels of LDL cholesterol, all the mice were dead, whereas if you had a mouse that had high levels of LDL cholesterol because it was not soaking up the LDL from the circulation, about 30–40% of these mice actually survived a dangerous lethal infection. This study I have quoted to several of my patients. This is called the Leiden birth cohort. So the town of Leiden, Netherlands. They took people who were 85 years of age. They found 1,000 people there. And these people were followed for 10 years. It's not a small amount of time, 10 years. You take people between 85 and 95 and you follow them for 10 years. And you say, I'm going to divide these people into people with high cholesterol, middle cholesterol, and low cholesterol. 250 or greater high, 200 to 250 middle, less than 200 low. And say, I'm going to find out whether people with high cholesterol die or people with low cholesterol die. What do you think is happening here?
Old age.
She's right. Being born is a fatal disease. All of us are going to die. So all-cause mortality was, not surprisingly, the highest in the lowest cholesterol group and the lowest in the highest cholesterol group. The higher the cholesterol, the lower the risks of you dying. Now people said that this is unfair because people with low cholesterol were already sick So you can't take this study. So the researchers said, okay, we'll take these people and we will eliminate all the deaths that happen in the first 1 year. We will count it after the first 1 year. And when they did that, nothing changed. The risks for dying from heart disease was about the same. It looks a little higher in the high cholesterol group, but there was no statistical difference. What about cancer mortality? People with high cholesterol had the lowest cancer mortality. Isn't that amazing? People with the lowest cholesterol had the highest cancer mortality. What do old people die of? Tell me, what do old people die of? So they die of infections, cancers. Lung infections are very common. So do you think that the highest cholesterol group had the lowest risk of lung infection, or was it vice versa? So the higher the cholesterol, the lower the risk of lung infection related deaths. I told you several things. I want to skip a few things because all I want to leave you with is that cholesterol is an important part of the cell membrane, and the LDL cholesterol is involved in repairing the cell. So let me say that again. Cholesterol is an important part of the cell membrane, and the LDL cholesterol is an important part of repairing that cell membrane. I mean, I can take you through this and show you how it does that, but I'll skip that because we want to get to some important points. Is there a level of cholesterol that is too high and that I would definitely want to treat? That is the question. The right answer to that is twofold. Are you talking about somebody with high cholesterol which is also poor quality? In other words, they have high triglycerides, low HDL, they are insulin resistant? That is different. But let's say I presume you are saying you have high cholesterol but high good quality cholesterol, right? If that is the question, my personal feeling is that the higher your cholesterol, the longer you're going to live, the smarter you're going to be, the better memory skills, the lower infection rate. So instead of getting worried about it, I would go and celebrate is the way I would think. I would do a CAT scan of the chest for calcium scoring, make sure you have no plaque so that you can reassure yourself. And that's the answer to your question. On the other hand, if it is poor quality cholesterol, then that's a different ballgame, if that makes any sense.